Dietary sugar is commonly in the form of sucrose, which is simply just glucose and fructose bound together by a glycosidic linkage. And dietary sugar is primarily digested within the small intestine. So sugar will enter the mouth, go through the esophagus, eventually enter the stomach, and then enter the first part of the small intestine, where this sucrose molecule right here will encounter an enzyme called sucrase on the brush border of the epithelial cells that line the gut. And so the brush border is simply just this little border right here that's shaped like a brush. Typically it's shaped kind of like this, where you have all these wave projections that increase the surface area of the gut to increase the likelihood that these molecules will interact with enzymes that will break them apart. And this sucrase enzyme will break apart sucrose into glucose and fructose monomers. So imagine this is a molecule of fructose now. This little blue molecule is free fructose. It was broken apart from sucrose. It's in the space within the gut, which is a space inside the body, but outside of an epithelium. This right here would be considered the epithelium. This would be considered interstitial space, and this would be our little capillary right here. And so this fructose molecule will move into the cells that line the gut, the enterocytes, through something called a GLUT5 transporter. So this is called GLUT, GLUT5, GLUT5, or the glucose transporter number 5, which is specifically kind of intended for the absorption of fructose. And it is insulin independent. So it doesn't require insulin to be present on the membrane of these enterocytes, or these cells that line the small intestine. And fructose will move through these GLUT5 transporters into the cells that line the small intestine. And then they'll go ahead and they'll move through a different kind of transporter called the GLUT2 transporter, GLUT2, into interstitial space. Oops, I used a different color. I'll use blue for fructose. And then fructose will then move into circulation. And here, this is just a capillary that's made up of a bunch of different cells. Each one of these is an individual cell, and those cells are called endothelial cells. And so this little fructose molecule is small enough to fit through the gaps between the endothelial cells and enter circulation. And then eventually it'll enter the hepatic portal vein. So it's moving through these capillaries. Eventually it's moving into venules, which are kind of small little veins, eventually into larger veins, which then coalesce into the hepatic portal vein, which goes straight to the liver. But it's important to note kind of what the affinity of these transporters are for fructose. So GLUT5, that's positioned on the very membrane of this cell that's facing the gut, which is called the apical membrane, has what's known as a Km, or a Michaelis-Menten constant, that's very low, which means that you can have not very many fructose molecules, and this still works really efficiently. So it has a really high affinity for fructose. Whereas the GLUT2, this guy, has a much higher Km, so you need a lot more substrate, you need a lot more fructose in order to get that fructose through these GLUT2 transporters. And so once we have fructose in these capillaries, it's moving towards the liver, a lot of these veins will kind of merge that are absorbing nutrients from the small intestine, and all that nutrients is going right to the liver, where the liver is the primary handler of fructose. And going back over here, it's important to note that in order to get fructose from the space within the gut, you need a relatively high concentration of fructose. So the dose matters in terms of the sugar content and the rate of absorption. So if I have sucrose, it takes a second, it's really quick, but it takes a second to break this little bond into glucose and fructose and then absorb fructose. But if I have free fructose in the form of high fructose corn syrup, that could potentially be more of a problem because the fructose is already free and there's a lot of it. So it moves through these GLUT5 transporters very quickly, which starts accumulating more and more fructose on the inside of these cells, which allows that increased substrate to then move through these GLUT2 transporters, with an increased concentration gradient, which then causes more fructose to move into these little capillaries, which eventually form the hepatic portal vein once they combine in venules and then enter the